Good afternoon, everyone, and welcome to this webinar presented by the Order of Australia Eastern Suburbs Regional Group. Lovely to see people here. Today, we're very pleased to be able to welcome Jenny Graves to present our webinar. I said to Jenny just a few minutes ago, if I said you're an evolutionary geneticist, does that capture it? And Jenny said, well, that just captures one little bit of it. That she has had many strings to her bow, so to speak. And she told me that she started as a molecular biologist and then moved through from there into a number of different areas. Jenny is a scientist with a long record of academic achievement, particularly, as I say, in the field of evolutionary genetics. Has been recognised in many ways. She won the Prime Minister's Prize for Science a few years ago and has a number of other awards to her name. Jenny, I'll let you introduce yourself in whatever way you think is appropriate in a moment, but we look forward to hearing from you and to what you have to share with us about a topic which sounds fascinating. I think we're all going to be educated in a very interesting way.
Well, thank you very much, Jeffrey, and it's a great pleasure to speak with you this afternoon. I thought it would be fun to talk about the work that we've done looking at the genes and the chromosomes that determine what sex you are, boy or girl, but particularly talking about the Australian animals that we've looked at that help us figure out how it works in humans and how it evolved. So I'm calling it Sex, Genes and Chromosomes and the Future of Men, and you'll see what I mean by the future of men, because what I'm going to tell you about is the X and the Y chromosome and what's happening to the Y chromosome. It's getting smaller and smaller, and it will disappear in several million years, and men get very worried what's going to happen to us. So what I work with is not just any old mammal like mouse, but I work with kangaroos and platypus and birds and snakes and lizards and frogs and devils and dragons. So I have something like 26 different animals that I've worked with over the last 40 years or so. And the reason what I do is to look at their chromosomes and look at their genes and where their genes map on the chromosomes, and more recently to actually look at DNA sequence of the genes. And the power of this is because kangaroos and platypuses and devils and dragons are very distantly related to humans. And that means they do things slightly differently, and it's differences that allow us to look to see what genes and chromosomes do. So I'll start off with a question you might not have asked yourself. Why is there sex anyway? Why are there two sexes? And why is sex almost ubiquitous in animals? Almost all animals, higher animals at least, have two sexes. And this is a puzzle, because why would you share your genes? Your genes would actually do better if you cloned yourself. So instead of sharing your genes with a partner and your offspring having only half your genes, you could clone yourself and have all your genes. And this has actually been a big question. And in fact, there are some animals that do clone themselves, or they make eggs out of their own genes. For instance, there's a little gecko which is female only. This is an Australian gecko, and there are only females in the population, and they make eggs out of their own genes. And so it's possible to do it. So why don't we all clone ourselves? Well, the answer seems to be that it's very good to have variation. And the way I explain this is, let's say there's a proto-giraffe with short legs and a short neck, and it can't reach the trees. So if there's a mutation that gives it a long neck, and another mutation that gives another population long legs, you get these giraffes, but they can't reach the trees either. Now, you could either wait several million years for another mutation to occur in the long-legged giraffe to give it a long neck, or vice versa, to give the long-necked giraffe long legs, but that would take millions of years. Way better for those two to mix up their genes and produce a really tall giraffe that can eat the trees. So generally, it's very powerful to be able to combine different variants of the genes, particularly if there are new evolutionary niches for a new species to go into. But probably the most, the best case for combining genes is actually confusing the pathogens. Because if a bacterium or a fungus can infect one gecko, it can infect all of the geckos. So you would get a real collapse of the population. But if all the geckos were genetically different, the poor old fungus would only be able to infect one animal. So it seems to be that mixing up the genes is really a good idea, particularly to keep the population healthy. And we know from many examples of animals that have kind of run out of genetic variation, like cheetahs, for instance, and Tasmanian devils, they're in trouble because the animals are too closely related. So that's why we have two sexes. Well, how does it work? Well, both sexes make gametes. The male makes sperm and the female makes eggs by definition. And essentially, you're just combining a sperm and an egg to make a new creature. So why are there differences between the sexes? Well, there have to be some differences because making a sperm and making an egg are somewhat physiologically different processes. But that's not all. You've also got to attract a mate. And also, for many animals, you have the function of caring for the young animal. So that means that there are some very significant sex differences. And for instance, you all know about peacocks. The male peacock has the most enormous tail and the female doesn't. Now, that's just because the male has to attract a mate, and that's how he does it, by having better feathers than the next male. And then a lot of lower animals like this mite, you have extraordinary sex differences. That lower picture there is a mite where the male is the little orange guy, essentially just sitting on top of the female. So when you look at sex differences between humans, they're not that significant. Basically, males and females have the same body plan. Males are a little bit bigger, and males and females have different genitalia, make different gametes, but basically we have the same body plan. We don't have a fan tail and we don't have huge size differences. So when I ask people, and I've done this for high school classes and the Royal Society of New Zealand, I've asked them to write down in 1 minute all the differences they can think of between men and women. And this is what Year 10 at Eltham College wrote many years ago, but it's almost identical to what the Royal Society of New Zealand wrote as well. What are the differences? Okay, well, males make sperm and females make eggs. Actually, hardly anybody mentioned that. And then there's a difference in reproductive equipment, and I have to tell you the penises and breasts won hands down. But secondary sexual characteristics are more subtle, but they seem more important to people. And oddly enough, the behavior, and just I just have to alert you to the fact I didn't write this. The Year 10s observed that their behavior were that boys are loud and silly and girls are bitchy and caring. But these are the perceptions that high school students and the Royal Society of New Zealand have of differences between men and women. So there are a lot of differences when you look harder. But the really important difference genetically is in the formation of testicles. If an embryo forms testes, everything else follows. The testes make hormones, and the hormones make the baby a boy. So really, genetically, it all comes down to making a testicle rather than an ovary. So that's the genetic event that makes boys and girls. So how do you make a testis or an ovary? Well, it's really interesting. If you look at a young embryo, it looks pretty much the same for fish and birds and humans. It looks very much the same. This is something like the 10th to 12th week of development in a human, but it's virtually the same in a mouse, but much earlier. And it's quite late in development. So you've already got the differentiation of the gut and the embryonic kidney and the backbone, but there's a little ridge of cells called the genital ridge, just a little bunch of cells there. And it hasn't decided yet whether it's going to be a testis or an ovary. But that's quite unique that you've got one group of cells that has two quite different fates. And so it depends whether that group of cells forms a testis with cords that make sperm, or alternatively, it can form an ovary that makes eggs. So this is the big decision. So what makes that decision? Well, there's a trigger, and the trigger can be genetic, and it can be either a male determining factor, or it could be a female determining factor. But some animals don't have genes and chromosomes that are different. It's up to the environment. And usually it's a function of temperature that the eggs are incubated at. And you probably all know that crocodiles do it by temperature. If it's cold, all the eggs hatch into females. If it's warm, all the eggs hatch into males. So it's terribly interesting because it's not genetic, it's environmental, and we're actually doing a lot of work about it, but I'm not going to talk about that today. So what I want to concentrate on is this genetic factor. So everybody is looking for what is the male determining factor, what is the gene, how does it work to make a testis? Well, the way we look at it, I'll just quickly show you what we do. We look at the chromosomes. Now, chromosomes in humans, we've got 23 chromosomes, different, and we have 2 copies of each, one from mother, one from father. Kangaroos only have 8 chromosomes, 2 copies of each chromosome. But it's the same genome. We have essentially the same amount of DNA, something like a meter of DNA, but it's just cut up differently into different chromosomes. So kangaroo chromosomes are actually lovely to work with because there aren't very many of them and they're really huge. In fact, they're so lovely, they actually made us a stamp out of chromosomes, out of kangaroo chromosomes in 2003. And I must say it was really a hoot working with stamp designers. They'd bring me up and ask me questions like, well, what color is a chromosome? I said, oh, I don't know. I suppose it's kind of a dirty white, but we stained them blue. So that's why you see blue chromosomes in my photograph, and that's why they colored them blue in the stamp. We have some lovely techniques that we can use to find out what genes are on these chromosomes, because of course the DNA is composed of a string of genes, and they're essentially the same genes in a human and a kangaroo, but they're just differently arranged. And we have some beautiful techniques where we can actually isolate a particular gene, so a bit of DNA, and we can tag it with a different chemical that fluoresces red or green or yellow. So you can see there that what we've done is we've looked at 2 genes, and we know that these genes are close together on the X chromosome of a kangaroo. And we looked at those same genes, on the X chromosome of a human. We looked at the same genes in a kangaroo, and we found that they're close together on the X chromosome as well. So we've also done a lot of work with platypus, and they have simply horrible chromosomes. You can't really tell one from the other. So what we did here is we could actually separate the chromosomes by size. So we could physically separate chromosome 1, which is the biggest, from chromosome 2, which is the next biggest, and chromosome 3, and so forth. And we could make DNA out of them, and we could tag it different colors. And this is called chromosome painting, and we use it a lot just to recognize individual chromosomes. But you can also use it to compare the genomes of different animals. And for instance, here we've used the chromosomes of one kind of kangaroo, which has got 16 chromosomes, and we've sorted the chromosomes, and we've tagged 3 different chromosomes, 3 different colors, and then we can use our paste to find out where those chromosomes are. In a different species, it's only got 10 chromosomes. And you can see that they're much bigger chromosomes, but essentially you're just jamming the green chromosome together with a red chromosome and the white chromosome at a tip. So you can see that the genome has rearranged between different species, but essentially it's the same genes in the same order, just differently, blocks of genes are just differently arranged. So that was an armory of techniques up in the 80s and the 90s, and then DNA sequencing came along. And DNA sequencing started off being incredibly expensive. They did the human genome first, and I was involved in that a little bit. That cost $3 billion. We were fairly early in it, and we did the possum genome in 2007, and that cost a mere $80 million. And by the time we did the platypus genome, that only cost $40 million. And we did the koala a couple of years ago, and that cost $10,000. So it's absolutely amazing that the technology has got so powerful and so cheap that we're doing $4 billion worth of work over 10 years for the human genome. And now we can do a genome in a week, and it will only cost $3,000 or $4,000. So this technology is incredibly powerful, and it allows us to do many things we could only dream of 20 years ago. Well, the secret of my work is comparing the genes and chromosomes in different mammals. So we compare chromosomes, the genes, and DNA sequences between different animals, and we do this to understand how sex works and how it evolved. So this is just my little diagram to remind you how different animals are related to each other. So there are humans and mice there. Everybody else works on human and mice because that's where the money is. But we work on elephants. And the reason for that is that elephants are just about as distantly related to humans and mice as you can get. We last shared a common ancestor about 105 million years ago. And so if we compare an elephant and a human, we're looking back in time to about 105 million years. But that's not far enough. And so we did a lot of our work looking at marsupial animals, that's kangaroos and their relationships. And now we're looking back a lot further to 148 million years, which is the last time that marsupials shared a common ancestor with us. And we could go back even further, looking at the egg-laying monitoring mammals like platypus and echidna. Now we're looking back 166 million years. And I kind of thought, well, that's enough for one lifetime. But some of my more adventurous students said, no, let's go back in further and look at birds and reptiles. Now, birds actually are a branch of reptiles, and they all are related to us by about 310 million years. So you can see the power of this. Now we're looking at the same genes and the same chromosomes, but we're looking way back in time. And we can do this with amphibians and even with fish. And now we're looking back 450 million years. Now, I'm not going to talk much about fish and amphibians in this talk. I'll concentrate on the mammals. So let's go to humans, because we know most about human sex. And you probably know that females and males all have 46 chromosomes. That's 23 from your mum and 23 from your dad. And we can tell which is which, not just by their size and their shape, but also we can create banding patterns in them using various cytological tricks. And so we can pair them up. And there you can see we've got 2 copies of chromosome 1 and 2 copies of chromosome 2. And in fact, in females, we've got 2 copies of everything. 1 copy from your mum and 1 from your dad. But males are a little bit different. You can see there's 2 copies of everything except 1 chromosome. And that was thought to be really strange. And that chromosome was called the X, not because it's an X shape, but because it's weird. X is for unknown. How come females have got 2 copies of this chromosome and males have only got a single copy? But that's not all, because males have also got a little chromosome called the Y chromosome to match the X, which is male specific. It's only in males. Okay, so there's a chromosome difference, and that means there's a genetic difference because the X chromosome's got about 1,000 odd genes on it. So we could say, right, well, here we have the chromosomes and the genes on it that determine whether you're going to be a boy or a girl. Well, okay, is it the number of X chromosomes, or is it the presence or absence of the Y chromosome? And people already knew about sex and fruit flies, that it is the number of X chromosomes. But that's not the case in humans, and we know that because there are girls who have only a single X chromosome, and yet they're girls. And there's boys who have 2 X chromosomes, but they also have a Y, and they are definitely boys. So that must mean the trigger for making a testis is on the Y chromosome. So this was known way back in 1959, that the testis determining factor, whatever it is, has got to be somewhere on the X chromosome. So we didn't know what it was, but we knew where it was. Now, this is going right back into the 1980s, when I wasn't working on the Y chromosome at all. I was actually working on the X chromosome, which is very interesting because it's got 2 copies in females and only 1 copy in males, which is obviously not fair. So I was looking at the difference in dosage of genes on the X. So I wasn't interested in the Y chromosome particularly, but I knew that there was a real war on trying to find the sex determining factor on the Y. So there's a blow up of our sex chromosomes. On the left, it's an XX in a female. On the right, a single X and this little chromosome called the Y in a male. So somewhere on the Y, there's a testis determining gene. Where is it? Well, people had been looking for a long time at patients who had parts of the Y chromosome, but not the whole Y. There's some people who have the whole of the bottom part of the Y, but they're missing the top part, and they are female. But there's other people who have just the top part and are missing the whole of the bottom part, and they're male. So that tells us that the testis determining factor must be somewhere in that little top bit of the Y chromosome. So we already knew by 1959 that that gene was very powerful because somehow it turned on other genes to make a testis. And the testis in the embryo makes hormones. And it's actually the hormones that determine whether the embryo develops as a boy or a girl. So what was it on that Y chromosome? We were very excited in 1988 or 1987 because a gene was cloned from the human Y. It was the first gene that was cloned, and it looked like a very good candidate for a testis determining gene. If this is so, it should be on the Y in all mammals. This gene was discovered in Boston, and I was very excited by this paper and told all the students they ought to read it because it's a beautiful piece of work determining this gene. And I was quite surprised when the guy who discovered it, David Page, called me up and said, well, if it's the right gene, it should be on the Y chromosome in kangaroos. Would you map it for me? So I said, oh, sure. And I had 2 students in my lab at that time, Andrew Sinclair of the Red Braces and Jamie Foster next door to him. I said, well, boys, why don't you go away and map this gene? We're pretty good at mapping genes in kangaroos. It'll probably be on the Y chromosome, and maybe you'll have a little teeny-weeny paper somewhere. So they went away and mapped it. And they called me up at 1 in the morning and said, well, I hope you're sitting down because it's not on the Y chromosome. It's on chromosome 5 in kangaroos. So that's a funny place to keep a sex-determining gene. It's the wrong gene. So I had to call up David Page and say, I hope you're sitting down because I'm sorry, but your gene is the wrong gene. So that's really the piece of work that convinced me that looking at kangaroos was a really useful thing to do. You could discover things that you can't discover just by looking at humans. So the boys got a fantastic paper and a cover in Nature that made them both very famous. And also, it gave them the opening. Andrew was actually the one who went to London to do his postdoc, and he was the one who cloned the right gene, which is called SRY. And Jamie was the one who found a gene on the X chromosome, which is actually the ancestor of SRY that I'll talk a bit more about, and another related gene that SRY actually interacts with. So this turned into a huge project, and it turned my life around because no longer was the X chromosome my focus, but I thought, well, it'd be pretty interesting to look at the Y chromosome as well, particularly in kangaroos. Everybody else in the world is looking at humans and mice. We'll look at kangaroos. So one thing we wanted to know is we know SRY is the right gene because mutations in SRY give you girls who have a Y chromosome but a mutation in the SRY gene. You can also take this gene and stick it in a mouse egg with 2 X chromosomes, and it turns into a male. So we know it's in the right gene, but we don't know how it works. We thought, well, maybe there's a few steps between SRY and what it does. Maybe it turns on other genes that make a testis. Maybe there are a few upstream genes that turn on SRY as well. Let's go out and have a look at them. Well, we thought it would be easy, and it turns out to be really complicated. People who all over the world have been looking at this pathway, and it's not a simple pathway at all. You can see that this bipotential gonad, that's a little genital ridge that I told you about, can either form a testis or an ovary. If there's SRY there, SRY turns on another gene called SOX9, that's the one Jamie Foster discovered, and that's the gene that turns on the testis differentiation genes. In the absence of SRY, it goes down a completely different pathway and activates other genes that turn on an ovary. But you can see it's quite complicated because there are other genes that turn off the female determining pathway, and other genes that turn off the male determining pathway. So it's kind of a push me, pull you. It's not really a pathway of genes, it's a whole network. And so far we know something like 60 genes in this pathway, all pushing and pulling towards making a testis or making an ovary. Now, one thing this means is that with all this pushing and pulling, there's a lot of variance. So there's a lot of children born who have a mutation in one or other of these genes. And those babies will often show differences in sex determination. So sometimes you get a baby and you simply can't tell what sex they're supposed to be unless you look at the chromosomes and the genes. So some of these are extremely trivial. There might be a baby with just a very short penis that has differences in the penile genes that make a penis. And there are other that are absolutely life-threatening. And if you have a baby with this gene, you must find out about it within a day or that baby will die. So there are many, many variants of sex determination, and it's a big worry in children's hospitals because it's a big worry what to do about them. Well, of course, that's only just making a testis ovary, and I'll tell you now a bit about the other variants. Oh, first of all, I have to show you my cartoon of what I think the pathway really looks like. It's probably something like this Rube Goldberg device where you have little balls going down, and if the ball gets into the genital ridge, it turns into a testis. But if it gets pulverized or smashed or inhibited in some way, and I love the little guy down the bottom putting it in a box and then emptying the box, you get an ovary. So this is a really good description of what's happening inside the biochemical pathway. It's extremely complex, and there's all sorts of ways of enhancing or suppressing that little blue ball. Well, we have gonads, testis, or an ovary, and the first thing they do in the embryo is make hormones, and that's really complicated. They either make androgens, which are male hormones and are responsible for male development, or estrogens. And again, there's a lot of genes that are involved in turning one precursor, usually cortisol, into any one of these hormones. And so again, you get a lot of variance of hormones. And again, this causes a tremendous lot of variation in sexual development that we know about. And then these hormones turn on physical development differences between males and females. And as I said before, basically females and males have the same body plan, except particularly for the genitals and whether the gonads make sperm or eggs, but also breast development, but also brain development. We know there are differences in the way genes are expressed in the brain. And so it's not surprising if males and females have different ways of using their brains. There was a study done a couple of years ago that really surprised people. They looked at what genes were active. We have about 20,000 genes. Males and females both have about 20,000 genes. And they looked to see whether they were differently expressed. They are both there in the genome, but a lot of them are differently expressed. They're expressed more in a male or more in a female. And to everybody's great shock and surprise, 7,000 genes of these genes are differently expressed in males and females. And not just in the gonads, not just in the breast, but in the liver, the kidney. Now you might think, well, why have different genes making a liver? A liver is a liver, isn't it? But there are differences in the way that males and females make livers and kidneys, and particularly brains. So I always have to show people this because a lot of people say, well, no, it's really only one gene that's different between males and females, so it can't be very important. But this one gene has extremely potent effects on hormones, and that has potent effects on physical development. One thing that gets people very upset is that there's clear differences in the way different genes are expressed in the brain, and so it is not surprising if there's differences in behavior. And the very interesting thing is that there are clear differences in genetic differences in gender identity. So there's a spectrum between male and female, and it's not necessarily aligned to whether you have a Y chromosome or not. So there are people who have a Y chromosome and they're definitely male, but they're born really believing that they are females. And so at the extremes of this, you do get trans, you get males who prefer to think of themselves as females and vice versa. That's not awfully common, but it definitely occurs, and it's probably got a big genetic component. The other thing is that, again, you get differences in mate preferences. So most males prefer a female mate, but a surprisingly high number of males prefer a male mate. And this is also very much influenced by genes. So they're definitely gay genes, which I prefer to call male-loving genes. And I think probably there's a very good reason why they are so common. That is that male-loving genes in the female relatives of gay men mean that they are likely to mate earlier and have more kids. So that's probably why these genes are so extremely common. So it's a very variable process making males and females, as you see. Well, back to sex chromosomes. Sex chromosomes are really unusual. I said that most chromosomes, you've got 2 copies, one from father and one from mother. They have the same genes on it, different variants of genes, but the same genes. That's not true of the sex chromosome. The X chromosome is a really big chromosome. The X and the Y look very different. They actually share a tiny little bit up the top with 24 genes on it. The X is a big chromosome, and there's probably more than 1,000 genes on it, and it's not dedicated to making females at all. There's many different genes on it. There's genes that make pigments. There's genes that make blood clotting factors. But there's way too many genes that are involved in intelligence. And we've known that for a long time because if you look at mental institutions, you would always find far more boys than girls. And that's because they have mutations in intelligence genes. And there's also way too many genes proportionally that have something to do with sex, either gonad development or fertility. So the X is not really normal, although it has a lot of different genes on it. And my cartoon of the X chromosome is that it's a very smart and a very sexy chromosome. Well, the Y chromosome is quite the opposite. There's hardly any genes on the Y, on the male-specific part of the Y. In fact, there's only 27, although there's many copies of some of these genes. In fact, most of these copies are inactive. So the Y chromosome looks extremely weird. There's lots and lots of repetitive sequence that's just got old, junky, old virus genomes in it. And there's all these inactive genes on it. The other thing is that many of these genes are needed for making sperm. So it does seem to be a chromosome which has been evolved to have many male-specific function. That, of course, includes the SRY gene, which we know does sex. So that's my cartoon of the Y chromosome. It's rather a wimpy little chromosome. In fact, as I'll tell you soon, it's actually a degraded copy of the X chromosome. So you have to ask, well, why are the sex chromosomes so weird? Is it so they work better? And actually, no, they don't work very well at all, particularly since the dosage of the X chromosome is so different in males and females. The alternative is it's a horrible evolutionary accident. So to understand the sex chromosomes, we need to understand a bit about evolution. And here's my picture of how the sex chromosomes evolved. This isn't originally my idea. It was known for fruit flies 100 years ago. And the idea is once upon a time, the X and the Y chromosome were just an ordinary pair of chromosomes that were the same, and they crossed over. That is, the bits of the chromosomes, the top bit could recombine with the bottom bit and form a new chromosome. And that's what my little dotted line is. until one day, one partner acquired a male determining gene. And we think that was the SRY gene. And that was okay, except that what it means is, by definition, this chromosome is now stuck in a testis. And what happens is that other genes, which are advantage in males, accumulate around SRY. Now, to keep these genes together in a sort of male-specific package, what happens is it stops recombining with the X. And so you get that whole part of the chromosome, which now sticks to itself. It's all isolated. And that's very bad for any part of the genome because it can't fix itself up. Ordinarily, if you get a mutation at one end or the other end, you can make one good copy of the chromosome by putting the two halves together. But you can't do that with the Y because it's all alone in the genome. So what happens is you get lots of deletions and mutations and a loss of active genes until you end up with a part of the Y chromosome is completely degraded. And that's pretty much a description of our X and Y chromosome now. Well, this can go on until the Y chromosome is degraded and degraded and degraded until it doesn't pair at all. And that's the way it is in marsupials. And it could even disappear. And it actually has disappeared in some rodents that I'll tell you about. So essentially, the Y chromosome is a degraded X chromosome, and this process is still going. You're still getting degradation of the X in different animals. So what does this mean for the genes on the Y chromosome? That's very interesting because this hypothesis suggests that the genes on the Y actually are copies of genes originally on the X chromosome. And these are just 3 that we happen to have discovered. They're partners on the X chromosome. And so one gene, RBMY, is important for making sperm. It has a friend on the X chromosome, which doesn't have anything to do with spermatogenesis, but is probably involved in mental retardation. Even SRY itself has a friend on the X chromosome. It's a very old gene. We find it's there in fish, it's there in frogs. It doesn't have anything to do with sex determination, but it seems to be involved in making a brain and making sperm. So it's quite interesting to ask, well, how did it ever become SRY? And so a long time ago, we suggested that what happened is you had this original SOX3 gene, which was active in brain and germ cells, and it got broken and rearranged, and it got expressed in the genital ridge. And we now know that's the case because there are some babies that were born that have no Y chromosome, but SOX3 is expressed in the genital ridge. And so what we suggested was there was a break here, a new bit of the genome, which had a sequence that drove its expression into the genital ridge, and that's what made SRY. So it's actually very easy to make new sex-determining genes. All we had to do was to express this old gene in a new place. Well, we went on to look at where did our sex chromosomes come from. We looked at the X chromosome, we mapped genes which are on the human X, and there's a human X and the human Y. We mapped them in elephants and kangaroos. What we found out was in elephants, it's the same genes in the same order. The only thing that's different is that little blue dot, which is actually the part of the chromosome that attaches to the spindle that pushes the chromosomes apart at division. But in kangaroos, to our surprise, we found that the blue bit, the bottom of the X chromosome, is the X chromosome in kangaroos, but the green bit is a part of chromosome 5. Now, what we found out was that actually these 2 bits of the genome are very old. We find them in chickens, We find the green bit is alive and well on chromosome 1. The blue bit is on chicken chromosome 4. They're not sex chromosomes at all, just plain old ordinary chromosomes. But what happens is that these two bits got stuck together, and that's why you have it in the elephant X, and the human X has just suffered a shift in this centromere. The other thing that's interesting is looking at the Y chromosome, you'll see it's mainly green. There's hardly any blue genes left. There's only 4 genes from the original Y chromosome, which is, of course, a degraded X. So again, you can see what we found out about by going far away from humans into elephants and into kangaroos and even chickens. We were able to give you the history of the X chromosome that started off just being an ordinary chromosome, had nothing to do with sex. 2 bits got stuck. First of all, the blue bit became the sex chromosome, then the green bit got stuck on it, and the Y chromosome was derived from that. Well, we found out even more by looking at platypus. And again, we're totally surprised because when we looked at platypus, we thought, okay, well, if we compare males and female chromosomes, we should find a couple of chromosomes which are different. There will be our X and Y. What we found out was actually there are 10 chromosomes that were different in males and females. And this is a very bizarre system. These 10 chromosomes actually link up together, and all the X chromosomes go into one sperm and all the Y chromosomes go into another sperm. So it's a very, very bizarre system. The really weird thing is that none of the X and Y chromosomes have anything to do with the human sex chromosome or the kangaroo sex chromosome. They're more like bird chromosomes. So it looks like if we look at the chromosomes, the sex chromosomes are down there at the bottom. They are clearly derived from something like bird sex chromosomes and have nothing to do with the sex chromosomes that we are familiar with from other mammals. Well, where are the human sex chromosomes and the kangaroo sex chromosomes in a platypus? This chromosome here is just ordinary old chromosome 6, is identical to the kangaroo sex chromosomes, and that's of course identical to the bottom half of the human sex chromosomes. So we've gone back and found that this chromosome is just an ordinary chromosome, not a sex chromosome, as recently as 166 million years. And again, that was quite a shock. It means our sex chromosomes are actually quite young. You may think that 166 million years is old, but just think we're going back 400 million years here. So if we look at the birth and the death of mammal XY chromosomes, if we look back as far as chickens and platypuses, what we find is our sex chromosomes weren't sex chromosomes at all. There were 2 different chromosomes, and they had about 1,000 genes on both copies of them. But then if we look back 148 million years to kangaroos, we find that the blue ones, the dark blue ones, have become X and Y chromosomes. If we look at 105 million years ago and compare ourselves with the elephants, we find that the sex chromosomes have joined up with the pale blue chromosome. And if we look at presence, we find that the Y chromosome with this new little bit has only got 51 genes left on it. That is the 24 genes that are shared with X, and 27 genes which are male specific. And what we can do is we can say, well, the poor old Y chromosome is degrading very fast. How long have we got left? Well, we can calculate that because we know it started off with about 1,000 genes 160 million years ago. So we can calculate that it's losing about 6 genes per million years, and there are only 51 left. And so we can calculate that if you come back in 8 million years, it'll be gone. Now, what's the future? Well, there's actually two lots of rodents that have gone through this already, and they have lost their Y chromosome already. So we can answer some questions. Well, what's going to happen? Will males disappear? Will we become like the gecko that clones itself? Will we become extinct? Or is there a possibility that we'll evolve new sex genes? And the good news is that these strange rodents that don't have a Y chromosome tell us that we can actually survive with different sex chromosomes. And I should just tell you quickly about birds, because they have completely different sex chromosomes and a different sex determining gene. So we're the weird animal lab. So we looked at emus. They have sex chromosomes that are identical, except the W chromosome, which is female specific, is almost the same as the Z chromosome. If we look at reptiles, we find again it's the males that have 2 copies of a big chromosome. The females have a single copy of that and a W chromosome with hardly any genes on it. It's a different chromosome, completely different. We found that out by gene mapping. If we look at lizards, we find many, many different kinds of sex chromosomes, both XY like humans and mice and kangaroos, and ZW like birds. So there's a great deal of variation. And if we look across the board, if we look right back into reptiles, so we can figure out what the chromosomes looked like 450 million years ago, we can see that the genome is essentially the same. It's just chopped up somewhat differently, but we can figure out that different parts of the same genome did sex in different mammals. So in snakes, the yellow chromosome became the sex chromosomes. In birds, the red chromosome became sex chromosomes. In platypus, it was basically the red ones, but then the aqua one got tacked on with a different sex determining gene. And marsupials started all over again with the blue chromosome and the evolution of SRY. Placentals are the same, except they'd suck the green chromosome on top. And so this is very nice because we can tell exactly when our sex chromosomes evolved, somewhere between 148 and 166 million years was when SRY evolved. So it's the same genome, but different regions determine sex. And so that's when SRY was evolved, and that's really quite recent in our vertebrate history. So again, I'm just telling you that Australian mammals were a big part of the story for why our sex chromosomes are quite new and how they evolved and how they work. Well, getting back to the weirdo sex chromosomes, we know that the Y chromosome is a degraded X, and we have to ask, well, why does it degrade? Chromosomes shouldn't degrade. Surely evolution should keep them nice and firm. Well, that doesn't work very well for the Y chromosome, partly because the poor old Y chromosome is always stuck in a testis. And there's many, many genetic accidents in the testis because there's a lot of cell division that you need for making sperm, and each one is an opportunity for a mutation. But the other bad thing is that selection doesn't work very well because there's only one Y chromosome in the cell. It's all alone. It can't repair itself. So that's why the Y degrades, and it degrades very, very fast. And as I said, we can figure out how quickly it degrades, and we can calculate that it's going to be lost in about 6 million years, or about 8 million years, depending on where you're starting. There it goes. The question of whether males will disappear, we hope we've answered. Are we going to form parthenogenetic females? No, we can't do that because we need sperm, we need males. There's at least 50 genes that are only active if they come from the male, and we can't develop without them. So yes, guys, we need males. Will there be new sex determining genes? And the good news is this has actually happened in some rodents. There's a little rat that lives in Japan on a group of islands. There's 3 species, and one of them has a perfectly good Y chromosome with SRY on it, but the other 2 have totally lost the Y chromosome. Both males and females have a single X chromosome. There is no SRY. Well, what else is there? Very recently, a Japanese collaborator has found out that just upstream of this very important gene, SOX9, there's a tiny little difference. There's just 17,000 bases, which is a very tiny amount, which has been duplicated. And this is what I've got as an EN. That stands for enhancer, and that's a control sequence, and it turns on SOX9. And if you've got 2 of them, it turns on much more strongly. So this little tiny duplication has now become a new sex chromosome. So it drives SOX9 even without SRY. You don't need SRY anymore. You've got plenty of SOX9 and you can make a testis. So yes, indeed, there's a new sex chromosome that we know how it works. So there's a network of genes. There's SOX9, a very critical gene, and it's become the new sex gene in the spiny rat. So who knows what's going to happen in humans? And it's interesting to speculate, well, what happens if a normal XX woman mates with a spiny rat man? Well, there's going to be a war of the sex genes here. And what's going to happen is that you're going to get a lot of infertility and a lot of intersexuality. And this is exactly the kind of barrier that will intervene and cause a new species to form. So this is exactly what's happened in spiny rat. There are 3 species and they're separated by having different sex determining mechanisms. So if you come back in 8 million years, you'll either find humans are extinct, or you may find there are several different species of hominids. So I don't know which is the more scary thought. I myself think we'll be doing very well to be on this earth in even 80 years, let alone 8 million years. So I think there are many other things that perhaps we need to worry about a bit more than the disappearance of the Y chromosome. So my conclusions then is that we have a lot of variation in sex-determining systems, and this is quite surprising for a very critical function that is very, very highly conserved. So what's conserved is the way the genetic network functions, but the trigger is very, very different. And in the humans, it's the SRY gene that does it. In birds, it's a different gene. In snakes, it's a different gene still. Looking at kangaroos and platypuses tell us that our gene is actually very young, and our sex chromosomes have been defined by this gene, and they're very young as well. It tells us also that the Y chromosome is degrading actually very fast, and it could be lost in just a few million years. And we have to worry about whether humans are going to become extinct or whether we're going to become different species. So I hope I've convinced you that weird animals, particularly weird Australian animals, tell us something about our own sex chromosomes that we wouldn't have found out if we just looked at human sex chromosomes. And I want to finish up by showing you pictures of all the people in my lab over the last 40 years who have either worked on platypuses or emus or snakes or dragon lizards, which is what I work on now. And I was really surprised when I calculated how many animals I'd worked with over 40 years. And it's something like 26 different animals, including fruit flies, kangaroos, dragon lizards, Tasmanian devils. So I hope you've enjoyed hearing about sex from an Australian mammal perspective.
Jenny, thank you so much. It's been a roller coaster ride, not only across the diverse animals that you've looked at, but across history as well. So thank you for that. We've probably got time for one or two questions. If there are people who'd like to ask a question, just give a wave, and then you need to unmute yourself to ask a question.
I think we're all stunned. Absolutely wonderful.
While people are thinking of that, I might also just mention that as well as being a world-famous scientist, Jenny this year has become a composer of opera as well. And some of you will know I'm associated with a choir called the Heidelberg Choral Society. And in the middle of the year, we presented a new Australian musical work called Origins that talks about the origins of the universe and of humanity from an entirely scientific perspective. And Jenny was the inspiration behind that. She came up with the idea and the framework, and worked with another member of the choir to put together words. And when we commissioned a young Australian composer to write the music, and it was wonderful. But that's a different story altogether. But it's certainly another feather in the cap of Jenny Graves. Yeah, Terry.
Is there any relationship with Dolly the sheep that was cloned in the sex? Was sex genes used for that development of the sheep?
Dolly the gene was cloned from cells that were taken from the uterus of another female sheep.
No.
So you would expect her to be the same sex as her clonal derivative, and that's always the case. You can change the sex in a petri dish by adding the Y chromosome, but I don't know that anybody's bothered doing that since we now know what gene does the job and vaguely how it works.
Jenny, I'm fascinated about how you determine the time frames. Could you say something about that?
Yeah, well, that's something that I really wasn't terribly interested in. You know, who cares if kangaroos are more closely related to wallabies and possums than they are to humans? But it's actually critical, and it allows us to tell when things happened in evolution. So we have pretty good evidence, both from the fossil evidence, but more and more from DNA sequence, we can tell which animals are more closely related. So humans are quite closely related to mice, very closely related to monkeys, much less related to elephants, and much, much less related to kangaroos. And so we can draw what we call a phylogeny, that is a family tree of all mammals. In fact, we can draw it right back to really the beginning of life. I've been involved in some work tracing these chromosomes, not just back 166 million years, but 730 million years. We shared a common ancestor with a tiny little lancelet fish that doesn't even have a backbone. But the chromosomes of that little fish are identical to some of the chromosomes of a chicken, which are identical to some parts of chromosomes of humans. So same genes in the same order, nothing has happened to them for 700 million years. I find that absolutely incredible. There's a big move on now that I'm a very small part of to get DNA sequence of all complex life on Earth, which is amazing to think of. And that means, you know, millions of species. But that will mean that we can tell exactly who is related to whom and when all these changes happened. And one thing you might not realize, it took me decades to realize it, that a lot of things in biology don't work very well. I mean, you think that we'd be just so terribly efficient because evolution would have got rid of all the mistakes. But that's really not the case with the products of evolution, which works in a very higgledy-piggledy way. You know, you don't invent new genes, you just work with what you've got and change things just ever so slowly. So sex is a wonderful example of how peculiar and weird and unnecessarily complex the pathways are. And that's probably true of everything. So don't think of yourself as, you know, a sleek convertible. Think of yourself as something with square wheels and band-aids and bits of string that hold us together.
There's a lot of accidents, and I'm reminded of one of the lines in Origins, which says that if you run the tape again, we probably won't finish up with humans.
That's right. And Drew Berry had a jellyfish that started, and the composer took us into another key. That was a wonderful moment.
Indeed. Jenny, thank you so much. It's been a fascinating presentation, and I'm sure that people have really appreciated that.